the last part of last online lecture concerned the different forms of neuronal injury, which are the neuronal acute swelling, characterized by swelling of the cytoplasm. Cytoplasm is pale with central or peripheral chromatolysis. The nucleus is marginated. This change is reversible. The second form is neuronal necrosis or quantitative necrosis. This is characterized by the cytoplasm is swollen, vacuolated, the cell membrane is not distincted, the nucleus is marginated and pycnotic and may show keratosis and keratolysis, and at the end the cell is resorbed or removed by the phagocytic activity of microglia. The third form is neuronal ischemic injury. In this form, there is complete chromatolysis. The nucleus is pycnotic and the cell is distorted in shape, and the cytoplasm is diffusely acidophilic. Such cell is dead cell. The fourth type is chronic neuronal injury. This takes place in chronic injury of the nervous tissue and characterized by distortion of the cell. The dendrites are tortuous. The nasal granules are come together, producing basophilic clumps. The spaces between these clumps are basophilic in staining. When the neuron is injured, it is surrounded by extra great number of microglia. The presence of such great number of microglia around the neuron, the degenerating neuron, is known as satelitosis or pathological satelitosis. Satelitosis is considered a main feature in pre-encephalitic stage. The normal neuron, there is usually few oligodendroglia, particularly in the neurons of the low cerebral cortex and the basal ganglia. This satelitosis is a process of microglia. When the neuron dies, the microglia receives this, they remove this dead neuron through its phagocytic activity in a process known as neuronophagy. Neuronophagy means the removal of a dead neuron by the phagocytic activity of the microglia. Now we finished the study or the description of the changes which can take place in the neuron body. Now we begin to study or explain or describe the change which can happen in the nerve fiber or the axon and the myelin sheaths. The injury of a nerve fiber can be indirect when the injury is found in the neuronal body, and in this case degeneration takes place in the neuron body and in the axon and the myelin sheath when is myelinated axon. Or direct, cut in the nerve. The upper part with the neuron body is found unchanged or have mild transitional change, while the distal part undergo degeneration. The degeneration begins usually by the axon, which shows swelling, fragmentation, and dissolution. And the myelin sheaths degenerate by undergoing swelling, fragmentation, and changing to or transforming to neutral fat or simple fat. When this happens in peripheral nerve, the process is called Wallarian degeneration. In this case, we find in the center at the site of cut center, fragments of the axon swollen and showing dissolution, while surrounded by droplets of myelin showing swelling and transformation to fat. This change is accompanied with very slow regeneration by budding at the cut end of the upper part of the axon of the nerve, depending on the proliferation of Schwann cells. In the blood vessels, perivascular, the blood vessels, the change in the blood vessels of the brain or the central nervous system قلنا ان perivascular cuff في inflammatory changes are common in the brain of the domestic animals, such as polyarteritis or periarteritis nodosa. But the non-inflammatory angiopathic changes or angiopathy changes such as atherosclerosis or medial sclerosis or arteriosclerosis are not common in animals. However, some typical lesions resembling these lesions could be recorded in some old animals, particularly dogs or oxes or the non-human primates. Now we finished the cytopathology and tissue reaction. We begin the changes which happen in the brain as an organ. We begin with the meninges. What can happen in meninges? There is metaplasia, osseous metaplasia in the meninges of old dogs and cats. The process of metaplasia begins from the frontal lobe, then parietal, then occipital. Also, other changes in the meninges are melanoma, menalosis. Menalosis may be local or a part in general. Menalosis is the increase in melanin pigment. The inflammation of meninges or meningitis, all types of meningitis or inflammation can take place in meninges except the catarrh. The lymphocytic meningitis is usually accompanying lymphocytic or non-purulent encephalitis, while the purulent meningitis can be as an extension from a nearby septic focus, such as cyanoadenitis in strangling in horse or purulent meningitis, purulent rhinitis, or purulent the media of the ear, the middle ear, or through blood, as in some specific diseases and listeriosis, like cirrhosis, and or septic emboli taking place. After this, we begin to speak about the brain oedema. The brain oedema may be inflammatory, may be non-inflammatory. It may be focal or general. It is found more frequently in white or more prominent in white matter than in gray matter. The accumulation or the brain with oedema is called also cerebral swelling or brain swelling. It is slightly swollen. The corrugation, gyri and alveoli are less distinctive. The cut surface is moist and glistening. Microscopically, the fluid or the water is found either perivascular or pericellular or in between the fibers of the nerve fibers of the white matter. A long-standing oedema may cause injury of the neuronal tissue or the nervous tissue, and also can produce gliosis, particularly the astrogliosis, because oedema is a stimulant for astrogliosis. Brain atrophy. Brain atrophy is not extensive in domestic animals, but it can be found in old oxes or in old dogs or old teeth. This depends upon the span life of the animal, how long this animal lives. The atrophied brain is smaller in size, have a color grayish yellowish in color, firm in consistency. When examined microscopically, there will be certain degree of decrease in the number of neurons, particularly of the cerebral cortex. And the neurons are distorted, more basophilic stains. The nucleus may be contracted. There is maybe increase in satelitosis. Also, there is increase in the glia fibrils.